Hello and welcome back. This is hello and welcome back. This is week 2 of the experimental biochemistry course, and we have a little more time left before it is 6 p.m. So we'll wait for the others to join. Okay, in today's class, we are going to talk about amino acids. So this course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and Professor Shomode from the Department of Bioscience and Biotechnology, IIT Kharagpur. And I am Bias Mukherjee, a research scholar from the Department of Bioscience and Biotechnology. And in today's class, we'll talk about amino acids, and we'll talk about the different classification of amino acids, what is isoelectric point of amino acids, how to determine the isoelectric point of an amino acid and a peptide. Okay, be it a dipeptide, a tripeptide, you will be able to determine the isoelectric point of this peptide by the end of today's class. Okay, besides that, we'll solve a lot of questions from previous year's NPTEL course and also questions from GATE Biotechnology course. Okay, so amino acids is a very important topic. It initially we'll start off with the basics, and then you will feel like it's very simple, but as we advance you'll realize that this is very important. And every year in GATE, in different exams, your different competitive exams like CSIR, NET, GATE, DBT, there will be questions from amino acids. Okay, and it's best to practice those questions. They are simple questions, so if you practice well, you will be able to answer them. Okay, so if you have any doubts regarding anything that we are discussing today or anything that we have discussed in the previous class, you are always welcome to speak out. Okay, so you can raise your hand and you can ask me the question directly. Otherwise, you can type in the chat box. If there is any doubt, any further doubt, then I'll give you the mail ID. Last class also I had given. So then you can mail me. Okay, we'll wait for 1-2 more minutes until it is 6 p.m. and then we'll start. So hopefully by then other students will also join. Okay. I've tried to make the PPTs very simple for you guys and also include as much information as I could. Okay, so if there are any suggestions regarding the presentations, regarding whatever I'm teaching or anything else, please tell me. Okay, I'm always in for improving things so that this course benefits you. Okay, so as new people are joining, let's start with today's class. We are going to talk about amino acids. Okay, first I'll admit everyone and then we can start. Okay, good. So all of you are coming exactly at 6 p.m. Very nice. Very punctual kids. Good. Okay, so hello everyone. As I was saying, in today's class, we are going to talk about amino acids. Okay, so we are going to talk about the classification of amino acids, isoelectric point of amino acids, and how to determine the isoelectric point of an amino acid of a peptide. Okay, so be it a dipeptide or a tripeptide, by the end of today's class, you will be able to determine the isoelectric point of peptides. Okay, and we are also going to solve a lot of hello, good evening to you too. Okay, so by the end of today's class, we are going to able to solve questions related to amino acids, be it MCQ questions or numerical questions. Okay, so there are a lot of questions from previous year's NPTEL course. There are questions from GATE BT, this year's GATE BT. Yes, good evening everyone. Okay, okay, good. All right. So let's start with today's class. Okay, so if there is anything regarding which you have any doubts, please write in the chat box or you can ask me directly. Okay, and if you have any suggestions regarding anything that I can improve in this course to make it more valuable to you people, you're always welcome to give in your suggestions. Okay, okay, let's start with today's class then. You tell me if you're able to see my screen. Okay. Right. So we'll talk about amino acids. Now what are amino acids? Amino acids are organic compounds that combine by peptide bonds to form proteins. Okay. Amino acids are amphoteric. What does that mean? That means that they are made up of ionizable, acidic as well as basic groups. Okay. So an amino acid has both an acidic group and a basic group. So the acidic group here is carboxylic acid, and the basic group that you can see in the figure, this is the amino group, alpha amino group. Okay. Therefore, they can act as both acids as well as alkali depending upon the pH of the media. That is why they are called amphoteric in nature. Okay. Now, amino acids can be of two different types. One is proteogenic amino acids and the other one is non-proteogenic. So what does that mean? So proteogenic amino acids are the amino acids which form proteins, and non-proteogenic amino acids are involved in other functions in the body but not formation of proteins. For example, the amino acids involved in urea cycle, right? There is citrulline, there are different amino acids. Those are very important but not important in forming proteins directly. Okay, right. So now interestingly, in the human body there are 20 amino acids, but in total in the world until now we have found 22 amino acids. So there are these amino acids selenocysteine and pyrrolysine, which are found in very special kind of bacteria only. Okay, not generally in other organisms. Now, non-standard amino acids such as gamma aminobutyric acid, which is important as the neurotransmitter in our brain. Okay, there is citrulline, there is ornithine. These are important in urea cycle. Okay, so they are very important in different metabolic pathways. Now, amino acids are involved in essential life processes, including building and repairing of tissues, enzymatic activities, digestion, assimilation of food, signal transduction, sensory perception, etc. Okay, now we'll move on. Now, amino acids can be essential amino acids and non-essential amino acids. So, what does that mean? So, from the food that we are eating, if we are getting some specific amino acids from our food, these are called essential amino acids. And there's amino acids that are prepared in our body by breakdown from different sources. Those are called non-essential amino acids. Okay, and some are semi-essential amino acid. That means that they are produced in our body but in very less amount. So those are semi-essential amino acid. For example, arginine and histidine, these are semi-essential. Rest, you can see that there are this list of non-essential amino acids which are made in our body and the essential amino acids which are not made in our body, and therefore you need to take these from outside. Okay. Now we'll look at the difference properly. So see that the amino acids, the essential amino acids are basically 7 in number and non-essential amino acids are 13 in number. Amongst them, one in adults is made in very less quantity, and that is of course your arginine, and 2 in children are made in very less quantity. Those are once again arginine and histidine. Okay, these are made in very less quantity in children, and that is why it's best to take from outside. Now, the essential amino acids, for example, glutamine in adult, this is made in less quantity when the body is in stress. Okay. Now, we come to an interesting question, which is, so, okay, no, we'll look at the functions of amino acids once again. We'll just brush through all these. So, one is breaking down of food, growth and repair of body tissues, making hormones and brain chemicals, neurotransmitters, then provide energy source, maintains healthy skin, hair and nails, okay, build muscles, boost your immune system and sustain a normal digestive system. Now, so that tells us that amino acids are very important for our bodily functions. Now, here is a list of different amino acids. This list is very long and it is made of 2 pages. Now, I will not be reading all of these one by one because that would be wasting your time. So, what you can do is, all these slides would be provided to you. Okay, you can go through this list. Even if you feel this is mugging up, even if you don't like it, I'm very sorry, but there might be match the following questions in your exams. So it is best to try to remember as much as you can. Okay, I wouldn't force you to do it, but it's best if you do. So there are different amino acids and their corresponding functions all mentioned in the list. Okay, so it's best if you go through. So this list is very long. There are 2 pages and I would recommend you to go through all the both the pages. Okay. Now we come to some interesting question, which is, so if now we understand that amino acids perform very important functions in our body, then the question is, do you think amino acid supplements are necessary for humans? Okay, so now all we have are these amino acid supplements and protein bars and so many different things. So all these extremely expensive protein bars and products. So the thing is that amino acids are essential for the body, but we more or less get all the amino acids from the food that we consume. But in case that, one second. Okay, so in case there are any deficiencies, then we can go for these amino acid supplements from outside. Okay, and normally this is all like provided by the food that we get. Okay, so deficiency of amino acids can cause headaches, weakness, irritability, fatigue, skin and hair related issues, insomnia, depression, loss of appetite, diarrhea and anemia. So this is what the marketing people tell you and scare you, and make sure that you buy all these amino acid supplements. Okay, right. Now we come to the different classifications of amino acids. Okay, so now amino acids are classified into different groups. So they can have non-polar side chain. So these non-polar side chain amino acids, they do not participate in intermolecular hydrogen bonding with their side chain, but they are involved in hydrophobic interactions and van der Waals interactions. Okay, now we come to polar uncharged side chains. These side chains are polar. So they have non-ionizable side chains that participate in intermolecular hydrogen bonding or ionic interactions. For example, serine. Now we were talking about selenocysteine, if you remember. So the interesting thing about selenocysteine is, in cysteine amino acid, there is this SH group, right? So there is this sulfur, and with sulfur H is connected. But in selenocysteine, there is selenium instead of sulfur. So if you remember the periodic table, then selenium and sulfur are very close by, right? Very similar. And that is why, very interestingly, in selenocysteine, this sulfur has been replaced by selenium. Okay. Now, another interesting thing is that if you read about crystallography, crystallographers use selenocysteine instead of normal cysteine for studying protein crystals, because it has more number of electrons, so it diffracts electrons very nicely. Now in this course also, as we proceed, we will talk about different forms of biophysical techniques, different types of biophysical techniques, and there we'll talk about crystallography. Okay, and then you can relate to this week 2 class. Okay, now we go to the charged side chain amino acids. So positively charged amino acids, for example, lysine and arginine, and the negatively charged amino acid, for example, glutamic acid, aspartic acid, these are all already charged. So of course they would be very active in intermolecular reactions and also intramolecular reactions. Okay, right. So you can remember this figure and try to. Also, once again, it's important that you memorize the structure of every amino acid. It will seem difficult at first, but it is very simple. All you need to do is just remember, and using logic, you will be able to get the structures. Okay, and even if you don't know the full structure, only the side chain you must know, because that is very important. Okay, so if we come to the amino acids that are having these uncharged side chains, then we will see that there are only 2 pKa values. So all these amino acids given in this slide, they are having only 2 pKa values. Now the funny thing is that for these amino acids, if you are asked to calculate the isoelectric point of these amino acids, then you have to just do only one thing, that is take the 2 pKa values, calculate the average or mean of the 2 pKa values, and the task is over. It's very simple. But examiners don't want things to be that simple for you. That is why you will be asked about the amino acids that are having more than 2 pKa values. So these are all charged amino acids. Okay, they are having 3 pKa values, and they'll ask you to calculate the isoelectric point of these amino acids. And how are you going to do that? So we'll talk about that in the upcoming slides. Okay, so why are there 3 amino acids? Because there are 3 ionizable groups. Okay, so one of the ionizable group is the COOH, that is one ionizable group. NH2, that is another ionizable group. And the third one is the R group, or the charged side chain. Okay, so that is why there is pK1, pK2 and pKr. 3 ionizable groups here. Okay, now there is a long list of different amino acids, the different pKa values of the amino acids, and there is the PI of different amino acids and the properties of different amino acids. Now interestingly, if you look at the amino acids which are neutral, they mostly have only 2 pKa values. If you go for the amino acids that are negatively charged, you will see that this R group is more or less greater than, it's very close to 7 basically. Okay, so the value of the pKr is high. And if you look at the amino acids that are acidic, so they are having very low PI, then you will see that this R group pKr value is close to the pK1 value of the COO minus group. Okay, and then I'm going to tell you a trick to calculate the PI of the different amino acids. So until now, if you understand this much, that's good enough. Okay, so as we proceed, I'll tell you more. Okay, now we'll start with some simple questions. So these are week 0 assignment questions from previous year's NPTEL same course, experimental biochemistry. Okay, you can try to answer these in the chat box. Okay, so now you have to remember the structure of the amino acids, you have to remember the 3-letter codes of the amino acids, and you have to remember the 1-letter code of the amino acid. All of these you must remember. Okay, so the question is, the correct 1-letter code for the tetrapeptide. Tetrapeptide meaning there are 4 amino acids. Okay, is, so it is tryptophan, asparagine, tyrosine and lysine. Option C. Okay, great. So very quickly you guys are answering. Very good. Yes, that is absolutely correct. It is option C. Very good. Okay, now we go to the next question. Now next question is the examples of amino acids with basic side chains at neutral pH. So basic side chain means that the side chain is going to have positive charge at neutral pH. So that means that these are positively charged amino acid. Which of these do you think are positively charged amino acids? Okay, try answering. Try. Tyrosine, methionine, glutamine, asparagine. Okay, so as we proceed, you will get to know more about the amino acids. No problem. Okay, so you are saying that it is A. Let's see. Okay, option D. So we have 2 answers. Yes, option D is the right answer. Why is that the right answer? You can see the figures here. Okay, so the pKa values of lysine and arginine are both given. Okay, now arginine has the highest pKa values for your this one for the side chain. Okay, so that is 12.5. Remember this, the examiners ask. Okay, so arginine has the highest pKr value and that is 12.2. So now interestingly, if you look at this table down, So the net charge of lysine at different pH is given. Net charge of arginine at different pH is also given. Why is that so? How is that so? I'll explain to you now. Okay, try to understand. So initially, when the pH is 1, then what is happening? The net charge is +2. Why is that so? Because this COOH group, this is remaining as COOH. Okay, NH3 plus is remaining as NH3 plus, and another NH3 plus, that is of the side chain, that is also NH3 plus. Now what happens when it is pH 3? You can see that the pKa of the carboxylic group of lysine is 2.2. So that means at pH 2.2, this COOH is going to give away its H plus ion. It is going to become COO minus. So what happens? There were two positive charges and now this one becomes a minus charge. So then your net charge becomes what? +1, right? Now what happens as you increase the pH more? At 7 pH, there is no change. It is still +1, right? At pH 10. So at pH 10, what is happening? You see that this NH3, alpha NH3 has a pKa of 9.0. So that means at pH 9.0, this NH3 plus is going to give away its H plus and it is going to form NH2. So it is uncharged. So that means there was one negative charge due to pKa and one negative and one positive due to pKr. So both of these together make 0 charge now. Okay. Then we go to, as we increase the pH further, at pH around 11. So see at pH 10.5, this side chain is also releasing its H plus ion. So then what is happening? These two are neutral and this one is having -1 charge. So the net charge becomes -1. Now if you increase the pH even more, net charge is still -1. Right? Now from here, how do you get the PI? That is isoelectric point. What is isoelectric point? It is the pH at which this amino acid is going to have net neutral charge. And it is 2 PI values, 2 pKa values that result in the 2 pKa values between the net charge 0. Okay, so here those are your pKa of the alpha NH3 plus and pKa of the side chain. So you have to take the mean or average of these two values and that will give you the final PI of that amino acid. So the trick is, for the negatively charged amino acids, the PI will be less than 7. So you have to take the two values that are less than 7. And for the amino acids that are positively charged, the PI is going to be more than 7. So you will have to take both the values that are more than 7. How is that? I have already explained. Okay. So based on whatever I explained for lysine, you apply the exact same principle for arginine and you will see that all the values that are given here are matching, okay, with your calculations. So that is how you determine the PI of an amino acid. Right. Now we will move forward and you will get a lot to practice. Okay. There are a lot of questions for you to practice. Okay. Now we go into the week 2 assignment. So we will start and you will try to answer. Okay. So a pair of amino acids containing aromatic side chain. So what are these aromatic side chains? Aromatic side chains are ringed side chain with this double and single bond. Conjugate rings are there, double and single bond, alternative double and single bond. So phenylalanine, okay, so you're saying that it is A. Good, yes, phenylalanine and tyrosine are both aromatic amino acids. That is absolutely correct. Okay, so all the aromatic amino acid containing, basically there are 3. We will not count histidine here. Histidine is not, we are not going to count this, okay, because there are not alternative double and single bonds. So we will only count phenylalanine, tyrosine and tryptophan here. All right. Okay. Right. So now we come to another thing which again you will have to remember, and that is the names of the rings or the names of the side chains. Now people will ask you, what is this imidazole group? What is an indole ring? Those you have to remember. Okay. Now different side chains of different amino acids have different names. So in this question they are asking you which pair has imidazole and indole rings in its structure respectively. Try to answer. Okay, so the options given to you are phenylalanine and tryptophan, proline and histidine, histidine and tryptophan, tryptophan and histidine. Okay, so indole, okay, C, okay, good. Let's see. Yes, that is very correct. Indole ring is this 5-membered ring of histidine that you see. Now, this is going to be very useful when we are going to study about affinity chromatography. Okay, and indole ring is this 6-membered ring along with this 5-membered ring joined together. That is the indole ring. And because of the presence of this indole ring, tryptophan gives immense amount of absorbance, okay, fluorescence. So it absorbs light at 280 nanometer and gives immense fluorescence. So that is something that you will remember. We have this in the upcoming slides, not of today's class, but later on. Okay, and then this will be handy then. Right, so we move on. Okay, now we are going to talk about something that is a little bit of stereochemistry. So what is this? So based on this Fisher projection formula, an amino acid can be chiral or achiral. So what is chiral amino acid? Chiral amino acid has four different chemical groups, 4 different functional groups attached to the alpha carbon. Okay, that is a chiral amino acid. Now some amino acids have more than one chiral centers. Some amino acid has no chiral center. For example, glycine, this has no chiral center. Why? Because glycine has hydrogen as the R group, right? So there are 2 hydrogens then. So there are not 4 separate chemical groups. Okay. Now this NH2 group can be either placed on the right hand side of the molecule or on the left hand side. If it is placed on the right hand side, it is called D-alanine. If it is present on the left hand side, this is called L-alanine. In our body, normally in humans and other organisms, maximum number of amino acids are L amino acids. In bacteria, there are some D amino acids as well, okay, especially in the peptidoglycan layer. So we'll talk about that soon. Okay, so L and D means describing the absolute configuration. This has nothing to do with the optical activity of that molecule. We'll talk about dextrorotatory and levorotatory very soon. Okay, so absolute configuration of a molecule that predates RNS. Now L and D have no relation to the optical rotation of a molecule. So when the amino group NH2 stays on the left side of the amino acid, then it looks like an L-shaped amino acid, and this can be thought of as the left hand. So your left hand. Whereas when the D-shaped amino acid are just mirror images of the L-shaped amino acid, so D-shaped amino acid is your right hand. Okay. Now we'll go to the next slide, and then we have this optical rotation. So what is this? Basically, light is made up of vectors of different like vectors are vibrating along different planes. Okay, electromagnetic, it's basically an electromagnetic radiation, and so the particles, they are actually the vectors are vibrating along different planes. So now what happens is when light passes through a polarizer. So what is a polarizer? Let's imagine it like this. For example, this is a slit and it is going to allow only the light carrying the electric and magnetic field vectors. So electric and magnetic field vectors always vibrate perpendicular to one another. Okay. So only the vectors that are parallel to this like orientation can pass through. So that is plane polarized light. So if you see the figure, you see that initially it was vibrating along, normal light has vectors vibrating along all directions. As it passes through the polarizer, it has plane polarized light. And when you pass that through a sample containing amino acids that are optically active, then they actually shift this light towards a different plane. That is called, so the plane of polarization of the light gets rotated. Okay, so all naturally occurring alpha amino acids except glycine. Why? Because glycine is not chiral, right? So chiral compounds have this property of optical activity, are optically active due to the presence of chiral carbon atom. They rotate plane polarized light. So D form or dextrorotatory form rotates the plane polarized light towards the right, and L form rotates it, L form or levorotatory form rotates it towards the left. Okay, now L and D forms of amino acids. Okay, so here it's written dextrorotatory for both. I will make the changes when I upload the PPT. Okay, please be careful. L is levorotatory and D is dextrorotatory. Okay, so L and D forms of amino acids are enantiomeric. What does that mean? They are mirror images, as they cannot be superimposed. So you cannot superimpose them when they are present in equimolar or racemic mixture. So they are present in equimolar or racemic mixture. Then the net rotation is null. Okay, because this is rotating it towards this is rotating it towards that, and the net rotation is thereby null. Okay, now if you look at this figure, that peptidoglycan monomer is given here, which contains N-acetyl muramic acid and N-acetyl NAG is N-acetyl glutamate most probably. So this has this pentapeptide structure, and there you will see that there are alternating D amino acids. Okay, so D-glutamate and D-alanine are found in the peptidoglycan layer in the cell wall of the bacteria. So this is something that you can remember. Okay, right. So now if I told you to remember the structures of the amino acids, because you will get question like these. So see, they are asking you which amino acids has the, which of these amino acids has the most number of chiral centers? Isoleucine, serine, tryptophan and threonine. Okay, you can try to answer in the chat box. Try to recall the length of these amino acids and which one has the most number of chiral centers. Okay, so I can give you a hint. Serine is a very small amino acids, right? So it cannot be serine. So you can remove the options containing serine. Then what are you left with? D. Okay, let's see. Yes, that is correct. D is the right answer. Okay, so isoleucine, basically isoleucine and threonine, both of these have the maximum number of chiral centers. Okay, and if you look at serine has only one chiral center. Similarly, tryptophan also has only one chiral center. But when you look at threonine, so threonine has two chiral centers, and isoleucine I think has one, and here, yeah, so it also has around 2 to 3 chiral centers. Okay. Now, which of the following amino acids has two chiral carbons? So this is a GATE BT question, right? Try to answer. GATE BT 25. If anybody has attempted GATE BT 25, you will know. Try to answer this question. Which one of these has two chiral carbons? So they are asking you which of these amino acids, okay, yeah, which of the following amino acids contain or contains. So that's a MSQ. It can be an MSQ, it can be an MCQ. So if you think one answer is correct, you can write that. If you think more than one answers are correct, you can write that. That is why these classes are so important. Chalo, try to answer. We answered this in the previous question. Chalo, try to answer. You know the answer. You just saw the answer few seconds back. C. Yeah, C is definitely there. So isoleucine has two chiral centers as we saw in the previous slide. And we also saw threonine. Right? Threonine also has two chiral centers. So both threonine, yes, that is correct. Very good. Yeah. So both threonine and isoleucine would be your right answer. Okay, that is why these practice questions are important, so that when you get actual GATE or NET or CSIR questions, DBT, etc. questions, you are able to attempt them. Okay. Now we'll talk about isoelectric point. So based on what we have discussed until now, I think it is already clear to you. So at physiological pH, most amino acids exist as zwitterides with both positive and negative charges. Right? The pH at which the amino acid has equal number of positive and negative charges is termed as its isoelectric point. The net charge on that amino acid is 0. Okay. Now, at pH values above and below the pi, the amino acid bears positive and negative charges respectively. How are you going to remember? It is exactly the opposite of number line. So, in number line, we know that on the left hand side of 0, it is negative, but on the right hand side of 0, it is positive. Here, if you see, when this is the pi, this is the pH value at which the net charge of the molecule is 0. On the right hand side of it, it becomes negative, and on the left hand side of it, it becomes positive. So at strongly acidic pH, the COOH group is retained, COOH is retained, and so the net charge on the molecule is positive. But at strongly basic pH, the COOH group releases the H plus ion, and the NH3 plus groups also release these H plus ion, and so the net charge on the molecule becomes negative. Okay, so a typical amino acid with neutral R group transitions from positively charged to a zwitterion and then negatively charged moiety as the pH increases. The zwitterion is a salt. So zwitterion is treated as a salt because it is having both positive and negative charges, right? So this is treated as a salt as a result of acid-base reactions of the basic amino group and the carboxylic acidic carboxylic group. So the acid-base reactions are as follows. Deprotonation. Deprotonation means loss of proton of COOH group as the pH increases. Protonation or gain of NH2 to give NH3 plus as pH decreases. Right? Understood? Okay. As you increase the pH, all the groups are going to lose H plus ions just in a specific order. As you drop the pH, all of them are going to accept H plus ions in the exact reverse order. Achha. So, hopefully you've understood that. Okay. Now we'll talk about Henderson-Hasselbalch equation. So, how is the isoelectric point of an amino acid determined? So, last class we talked about this particular equation, which is called Henderson-Hasselbalch equation, and pH is equal to Pa in this equation, and the quantitative measure of the strength of an acid is pKa, right? When the concentration of the acid or HA is the same as that of its conjugate base. That much we know from a previous class. Now lower pKa values mean that it is a stronger acid, and the pKa is the pH at the midpoint of the flat buffering zones in a titration plot, or pH versus volume of titrant. Okay, so if you look at this figure, you will see that the positively charged form dominates in lower pH, negatively charged form dominates in higher pH, and the region between is actually the neutral or the zwitterionic form dominates. So your Pi will be the 2 pKa values that are on either side of the zwitterionic form. Okay, So the isoelectric point or PI lies between the points A and B where the concentration of the acid is same as that of the conjugate base. Okay. Now we'll move on. Now we have the example of an acidic amino acid, a neutral amino acid and a basic amino acid. So if you look at the acidic amino acid, you see that its PI is always the sum of the two pKa values that are less than 7, and for the neutral amino acid there are mostly 2 pKa values only, so you get the mean and from there you get the net PI. For the basic amino acid you have to take the 2 pKa values that are higher than 7 and you will have to make a average of that, and that is how you get it. Why that is happening, that we have discussed. So I hope you understood that. Okay. The calculation of the PI of a neutral or a diprotic amino acid. What is a diprotic amino acid? This means that there are only two protonation groups involved. So be it COOH and NH3 plus. So there are only two chemical groups that are involved in loss of H plus ion when the pH increases, right? So average is pKa1 and pKa2. However, to find the PI of a triprotic acid, so there are three H plus leaving groups, H plus donating groups. So the average of the pKa values that are closest to the pH at which the amino acid assumes 0 net charge is used. Closest to the pH at which the net charge is 0. So we'll practice a few questions to make it simpler for you. Okay. So which of the following statements is or are true for the isoelectric point? A is the pH where the molecule carries no net electric charge. pH where the carboxylic group is uncharged. pH of the maximum electrolytic mobility and the field point. Okay. Which are true for the isoelectric? Okay. So you are saying that it is A. Okay, let's see. Yeah. Oh, sorry. I revealed the other question's answer also. So for the next question, which of these amino acids have PI close to that of the physiological pH? Okay, try answering. Which one of these are not charged? Which will have only 2 pKa values and only then they will have the PI close to the physiological pH. We have new people joining. Try answering the second question as well. Okay, so you are saying that the second question is answer is D. Okay, let's see. Right. So for the first question, what is isoelectric point? Isoelectric point is the pH at which the molecule carries net zero charge. That is true. That we understand. Good. Now we look at the other options. Okay. So the pH at which the carboxylic group is uncharged. That is wrong. Because if the carboxylic group is uncharged, that means the PI is the pH is very, very low. Because carboxylic group has a PI of around 2 point something. So if it is uncharged, that means the pH is around 1 or so. So that is definitely wrong. What about maximum electrolytic mobility? If a molecule is having 0 charge and if you apply a electric field, do you think it will move? It wouldn't move because in an electric field, the molecule moves when it has a lot of charge, right? So that is the principle of your SDS page and everything. We'll talk about that later. So if it is having no charge, it will not travel towards the oppositely charged electrode. So that means the pH of the maximum mobility electrolytic mobility, that is definitely wrong. It is the pH of the minimum electrolytic mobility rather. Okay. And about field point, this you will tell me in the next class. Okay, this is homework for you. Okay, you will tell me what is field point. Right, now we come to the next one. So which of these have pH closest to that of the physiological, which of these have PI closest to that of the physiological pH? So histidine is the amino acid that has the PI closest to that of physiological pH. If you look at all the other, all of them are having more than 1 pK values. Okay, and you can go back and look at these amino acids over here. 1 second. Yeah, so all of these different amino acids are here, and you can go through the PI of these different amino acids. Okay, so be it glutamine, be it threonine, you can check which of these are having PI values closest to that of physiological pH of 7, and then we can discuss. Okay, now we'll go to the next question. Okay, now histidine, it has its PI closest to that of physiological pH, and that is why this is very important in different enzymes. Okay, it is often there in the catalytic pocket in the enzyme, and it is very active in interaction with ligands, other proteins, etc. And in fact, depending upon the charge of the amino acids, the conformation of the pocket active site of the enzyme can even change. So an enzyme may be active at one particular conformation, but it can be not active at another conformation. Right. So that is why histidine is very important in this regard. Now we go to another question. So when amino acid alanine, which has R group of CH3, it is added to a solution with a pH of 7.3. Okay. Then alanine becomes what charge? So at a pH of 7.3, what charge do you think alanine would have? So it's okay. You're saying that it is A. Great. Let's see. Okay. So we have two different answers. Let's see. Okay. Acha. So C is the right answer. Why is that so? Because you see that alanine is having pKa1 and pKa2, and as we know that it is not having any charge for its R group. So pKr is 0. So that means always remember that this OH group has a pKa of around COOH group has a pKa of around 2, and this NH2 group has a pKa of around 9. So if you do the sum, it will come to be around 7. Okay, so if you see that at around 7.3, it is going to exist as a zwitterine. So both positive and negative charges are going to be there. Okay. But when you exceed pH 9, it is going to have only negative charge. When you go below pH 2, it is going to have only positive charge. Okay. Right, and its isoelectric point is around 6 based on that calculation. Okay, now we have another question. See, the PI of the following amino acid, pKa1 is 2.16, pKa2 is 9.06, and pKr is 10.54. Calculate the PI. I have told you a trick. Use that. So your calculations will be faster. Okay, calculate the isoelectric point of this particular amino acid. By the way, what amino acid is this? Anyone who can tell me? A. Okay, great. Tell me the isoelectric point of this amino. Tell me the name of this amino acid, if you can. Yeah, very good, correct. So the net charge, the PI of this amino acid is 9.8. Very good. Okay, and the name of this amino acid is lysine. If you go back to our previous slides, you will see that this is actually lysine. And why it is very significant and why is it very easy to understand that it is lysine? Because lysine has this NH3 plus attached to its epsilon carbon. So that means that alpha carbon has all the other functional groups, and then there is beta carbon, gamma carbon, delta carbon, and finally to its fifth carbon, this NH3 plus is attached. Okay. Now, we have something very important. Which of the following is or are polar amino acids? Now, polar amino acids are often there on the outer side of the protein because these interact with water molecules and they improve the solubility of the protein. Okay. Now, if the polar amino acids are very few on the outer side of the protein, then what happens is the protein will form hydrophobic interactions. Okay, okay. So you're saying that it is C. Acha. Okay. So you guys are saying it is C or D. And both of you are correct. Both are right answers. Right? So that is an MSQ, multiple select question. Okay. Right. Now we have two other questions. Anyway, so I was talking about these polar amino acids. So when the polar amino acids are not very abundant on the surface of the protein, then the solubility in water decreases for the protein and the protein will bind to other protein molecules and it will form this big polymer and it will eventually possibly precipitate or something. Okay. And those proteins are a nuisance in the lab. Anyway. Okay, so we have another question here. I've already got the answer, but I'll read out the question for you guys and you guys can answer the second question until then. So the PI of an amino acid having the first pKa 2.14 and the second one is 9.73. Okay, very good, very good. So you guys have understood, you all are answering very fast. Good. Right, so for the second question, it is during the pH titration of glycine with NaOH, a certain amount of HCl is added at the beginning. The role of HCl is, right, so we have a lot of answers. Let's see which are correct. So D and C. Okay, let's see. D and C. Okay, yeah, that is very correct. D and C are the right answers. So PI, you know, very direct, just the average. Very good, done. Now we come to the next one. What is the role of HCl? Completely protonates glycine. So it's a pH metric titration with NaOH. So that means we are going to decrease the pH gradually, right? So it is going to completely protonate glycine initially. So glycine when taken in water, it has a zwitterionic form. But during titration, equimolar amount of HCl equal to the amount of glycine present is added so as to protonate the glycine completely. This will allow us to determine the initial ionization pH of glycine, which is pKa, which would not have been possible if the zwitterionic glycine was there. Right? So glycine is becoming COOH, and then as you gradually increase the sorry, 1 second. Okay, yeah. So then as you change the pH, the titration curve is going to form. Okay. Right. Now, the answer is already given. Okay. We'll at least look at the question. So, this is the titration curve. Okay. Equivalence of OH. So, that means OH is added and the pH is increased. So, initially it is having COOH group, then as you keep on increasing different, first pK1, then the pK2, then the pKr, the different groups. This is once again which amino acid? We talked about this. Tell me, you guys know. Which amino acid is this? See, there are 5 carbons. So this is lysine once again, right? So when NaOH is added to a solution of lysine, drop wise, the number of inflection points. So inflection is this. Inflection points, that is sharp changes in pH observed are. So how many sharp changes of pH are observed? Tell me. How many inflection points are observed? So the right answer given here is 2. Okay, you can double check once and then we can discuss about it in the next class. Okay. Now, the next question is the pK2, that is pKa for the side chain. So the pK2 or pKa of the side chain of histidine is 6. That we know. That is why histidine is so important in biological processes. So when it is present in an isolated state, considering a specific proteins with histidine at its active site, the pKa is expected to be less than 6 if the active site has more positive charges. Great, Acha, you're saying that it is C. Okay. So greater than 6 if the active site has more positive charges. Less than 6 if the active site has more negative charges. And same as the isolated histidine, that is 6. So we look at the right answer. The right answer is actually A. So when histidine residue is present in its active site, having more positive charges, the pK2 becomes lower than 6 because positive charges in the active site destabilize the positively charged histidine side chain. Okay, so a higher concentration of protons, that is lower pH, is required to protonate this histidine side chain, resulting in lower pK. Got it? Okay, that was something interesting that we got to know. Okay, right. So now we will talk about a tripeptide. Can you please explain this? Okay, sure. So see what is happening here. They are asking you that, let's say that histidine is there in an active site, and in that active site there are also other side chains, okay, other amino acid side chains, and they are asking you that in the presence of these other amino acid side chains, will the pK2 of the histidine side chain still be 6 if it is not in the isolated state? Okay, so pK2 here is actually that of the side chain, right? So pK2 here is referring to that of the side chain because the side chain pK is only 6. Now, when the histidine residue is present in the active site having mostly positive charges, the pK2 becomes lower because positive charges in the active site destabilize the positively charged histidine side chain. Histidine is positively charged when it is like it is losing this, it is normally having this NH3 plus ion, right? So when there are a lot of positive charges, then it is basically affecting this side chain of histidine, and so higher concentration of protons, that is lower pH, would be required to protonate this. So this is already having very strong positive charge and you would require higher pH to make it lose this H plus. That is why lower pH basically to make it lose this H plus, and that is why lower pKa would be there. So lower pKa means stronger acid. So because of this, very like it is becoming more acidic due to the presence of these other H plus ions. Okay, hopefully you have understood. Otherwise, you can read more about this. Okay, if there are any other doubts, you can read about this properly. Okay. Right, so let's continue if that's okay. Next slide, sure. So now in the next slide, we have something that is a tripeptide. Okay, so until now we have been talking about single peptides. Now we are going to talk about a tripeptide. So for a tripeptide, you would be in fact worried that we have so many pKa values, right? So then which two would contribute to its pi finally. So what you have to do is you have to take into account all the pKa values, okay, all the ionizable groups. So if you look at this, you see that there is this aspartate and then there is this glycine and lysine. So net ionizable pKa are 4. Total 4 pKa values. Now what we will do is we will take into account the different pH, okay, and we will try to calculate the net charge of these amino acid of this tripeptide at different pH. So at pH 1, what is there which so at pH 1 both pKr and pKa1 they are going to be COOH. So the net charge would be positive 2 plus, right, because of this pKa2 and pKr, right, of lysine. Now what we are going to do is for pH 2.5 or let's say around 3, pH 2.5, what is going to happen? This pKa1 of lysine, this is going to become COO minus instead of COOH. So then we get 1 negative charge and 2 positive charges there initially. So we get plus 1 charge. Next, after pH 3.65, so around 4 or so, and then until 7 and then until 9, right? So from 4 to 9, what you will get is this other pKr of aspartic acid. This is going to lose its H plus ion. It is going to form COO minus. And so now we have 2 minus charges and 2 plus charges. So now the net Pi is 0. Next, as we increase it further, this pK2 of aspartic acid, this is going to lose the H plus ion. And if you increase it even more, around 11 or so, 10.5, then this pKr of lysine is going to lose its H plus ion. Then it is going to become minus 2 at pH 12, right? So now we will take the 2 pKa values that result in the formation of this zero charge. Which are these two? You tell me. Okay, you guys tell me. From here we can see okay, you can answer in the chat box. So look at this very carefully and you tell me which pKa values would you take into consideration. That is finally resulting in pH of 0. It is written on the slide. You people are not looking. Okay, so it's written on the slide. Yeah, right. 9.6 and another one is 3.65. The two pKa values that result in the formation of 0 pH basically, right? Sorry, 0 charge at pH 4 to 9. Okay. So the isoelectric point would be the average of these two values and we are getting that as 6.6. So this is how you calculate the Pi of a tripeptide. Now I have a question for you. This is a tripeptide. Oh, 1 second. Okay, so the answer is already given. Very unfortunate. Otherwise, I would have told you to do it. Okay, so there are 3 pKas. Oh, this is not a tripeptide. This is a normal histidine. So anyway, there are these 3 values, right? 3 pKa values and you have been asked to calculate the isoelectric point. And so if you take into account the values and the 2 different values between which we get 0 charge, then we will see that the Pi is coming to be 7.6 and this is the closest to neutral that we have, right? Okay, now we have another question. Try to answer this. So titration of arginine residue present in a protein is very difficult to carry out with NaOH unlike individual arginine. Why is that so? Okay, the different options are: protein forms complex with NaOH, multiple amino acids may make the process complex, D. Okay, the structure of the protein may degrade at higher pH, and D is all of the above. Okay, so we look at the right answer. excuse me. So B and C would be the right answer. Okay. So extreme pH can degrade the protein structure. In addition, it is not possible to estimate the PI of particular amino acids due to uh, because in a protein has multiple amino acids which can affect the measurement. Okay. Now that is the end of our chapter. Okay, so all the different references are given for your practice. Now I have marked these three important references. One of them is this virtual lab program. So if you see in my previous class, I had this. Wait, I'll actually show you. Okay, give me a second. I will share my full screen and then you'll be able to see. Okay, this would be very helpful for you actually. So hopefully you guys can see. So if we go to this one, okay, it's taking time to open. Yeah, so this is the one, right? If you guys can see, so this is the one. So it's a virtual lab training program. So normally what you do in a lab, the same thing has been replicated here, okay? And here you're doing everything exactly the same way but virtually. So if you look at this, it's very interesting. Sorry, one second. Yeah, so it's very interesting. You can see, I'll minimize this so that you can see properly. Okay, so this is actually a free website for you people. So pH titration of amino acids and small peptides and estimation of isoelectric point. So we all have developed this, and you can see that there is this aim. It's just like an experiment. Okay, so there is this aim and then objective, what all you can learn from this experiment, that is given, and then theory. So whatever we have learned until now, all of that is given here too. I have made this material. So yes, a lot of things that you have learned until now are given here. Okay, and something very interesting is also there. So how to calculate these dipeptides, tripeptides, PI, all of that is given here. So you can go through that. Next is applications. Application is normal. You guys already know that in different industries, in different sectors, these amino acids and their PI is very important, right? For example, protein purification and separation that we do in the lab and then people do in different industries, then forensics and disease mechanism, etc. Now we come to something interesting, and that is pre-test. So what is pre-test? This is actually a list of MCQs. Okay, so you can answer like this, and then they will, so you will have to go and submit, and then they'll give you the right answers. Okay, so these are additional MCQs for your practice. So you can try these. And then we have the procedure section. So here is the nice part. So see, there is this diprotic acid alanine. Diprotic means that this is having two jumps, right? So in the pH, and in between is the buffer zone, which is your PI region that is lying there. So you see that the PI region is around 6.1 and the pKa1 value and pKa2 values are given, and here they will ask you pipette 10 ml of 0.1 molar amino acid solution into a conical flask. So you will actually have to do that. This is the setup, and in this conical flask you're supposed to add amino acids, and then the pH meter is going to measure that. So to you can press control plus 5 to clear out this things. Okay, if there is any technical glitch, you can use control plus 5 to refresh, and then whatever is asked here you will follow that, and then you will be able to know how pH titration is actually done. So it will be very helpful for you to understand. Okay, and this is the simulation part. So simulation is where you will have to do it. So all the instructions are given here. You will have to follow that. You will start the experiment, and this is the view it, and you see that different things will be done here, and then here you will get the plot, okay, for the different amino acids. And then from here you can also get the PI, so you'll have to write the value, you have to calculate and give the value, and then they're going to show you the right answer for it. Okay, and in part B we have this tripeptide that we already practiced. So you can practice here, you can write the net charge at different pH and calculate the PI, and then you can check and it is going to give you the right answer. Okay, so that is how you can do it. And then we have post test again. So in post test we again have a lot of questions, and you can try to answer these questions. Okay, these are all MCQ questions, so you have to submit the quiz and they will show you the right answer. Okay, and then finally, one second. Yeah, so that's it. References and contributors are not necessary for you. So this is one thing that you can do. Okay, and we also have more questions here. So in this this particular link and the next link, there are a lot of questions, a lot of MCQ questions. Okay, you can actually go through those and you can learn more about amino acids, and I would recommend that you practice these MCQ questions, okay, as nicely as you can, because MCQs on, please send details of the virtual lab website. Yeah, it's there in your, okay, wait, I'll share the link here also, but it's actually there, okay, in the PPT. I'll share the PPT, that then you can get it from there, but here also I'll share it. So in virtual labs program, there are a lot of different experiments, and a lot of those would be useful for you. So I'm sharing this particular amino acids class part. Okay, one second. Yeah, so that's it. That's all for today. Thank you for your patience. Thank you for joining the class and being with me all this while. Okay, so I would recommend that you can go through these different websites that we saw. Okay, these would be actually helpful. There are a lot of MCQs, and the best part is if you go online and search, then there are MCQs on amino acids from different exams. Okay, they are given separately, GATE, BT, GATE, Excel, etc., etc. And like for example, this entire GATE paper is given for IIT Roorkee, IIT Kanpur. So whichever IIT made the GATE paper of that particular year, that is given. So I would recommend practice from there. Okay, a lot of questions would be there that you would not be able to answer because we have just started with the course. But try the questions that you can answer. Okay, as we proceed, you'll be able to answer more and more questions. Okay, so that's all for today. Thank you so much for your patience and participation. If you have any doubts, okay, I'll give you my mail ID once again. Last week also I've given you my mail ID. So if you're having it, well and good. A lot of people were not there in the last class. So you can have this mail ID. If there is anything that you want me to talk about in the upcoming classes, you can mail me at least one day back, because I would also need some time to know what to like change in the slides and what to talk about in the next class. Okay, so this is my official mail ID submitted to NPTEL. You can also have it. If there's anything that you want to communicate with me, any suggestions, any doubts, anything that you want to tell me, you can mail me there. Okay, that's all for today's class. Thank you so much. If there are no more doubts, we can end the class today. Okay. Okay, so since you guys don't have any more doubts, let's end the class. Yeah, thank you so much. Thank you to all of you. Okay, we'll see you again next week at the same time, right? Okay, thank you so much. Bye bye, everyone.